Welcome to Science Thursdays here at the museum. This is the Daily Planet Cafe, and it's our Science Cafe here at the North Carolina Museum of Natural Sciences. And we do this every Thursday. Is anyone here for the first time? Thanks for coming. All right. How did you? Okay. Well, we don't have to get into a big discussion, but thanks for coming for the first time. How did you hear about us? In the newspaper. That's awesome. It's news. You read the newspaper? Oh. So you're one of those rare people. So you don't get all your everything online. Well, excellent. We do this every Thursday, or we do something like it. In fact, next week is the first Thursday of the month in March, and we always do science trivia on that night. And for those of you who comes to our science trivia night, okay, cool. I'll be hosting. Paul will be out of town, so I'll be hosting next Thursday. And then we also have another spelling bee coming up in the middle of the month, March 24th. That's nowhere near the middle. And we have a couple really cool cafes next month. One is about nicotine addiction and the brain, and another is about, we've been having kind of a series of cafes featuring people from our vet school at NC State and our veterinarians, and this one is about injuries in horses and also applying the knowledge from that to human medicine. So that'll be an interesting one. But tonight is one we're very excited about. It's something that I'm really interested in, and I've taken part in some of the studies that we're going to hear about tonight. And our guest is going to talk for a certain amount of time. And then she also brought some students that are also going to be part of our program tonight. So that's why we have this big cast of characters. So let's start with our very special guest. She's our own director of our genomics lab up on the 3rd floor in this Nature Research Center. And she's also a professor of biology, biological, and biomedical sciences. All right, I should have just said professor. I tried. I thought I could pull it off, and I couldn't. But she's a professor at NC Central. So how about a nice round of applause for Dr. Julie Horvath.
Awesome. Thank you, Brian, and thank you all for coming out tonight. So I'm going to go through, I have some slides as backups, but what you guys all have on your tables. So one of the other awesome scientists in my lab, Sarah Council, actually handed out some pie charts for you. And so we'll talk about those a little bit later, but it's just everybody should have pretty much a different pie chart from a different person or an average of people. So we can talk about that later, and you can ask questions about that, hopefully. All right, so I have these 3 lovely ladies up here with me, and these are sort of the next generation of scientists. So they're working with me, doing some research with me, and so at the end I hope you'll hear a little bit about what their experience has been, and you can ask them questions as well. All right, so today we're going to be talking about armpits, and I'm going to start out just talking about microbes. So who knows what a microbe is? Most of you guys? Okay, awesome. Ooh, let's go back. So we have a lot of different things that we call microbes. These are microorganisms, so tiny microscopic life, right? And so there are different kinds of organisms that we classify as microbes. So we can have different bacteria, fungus, protozoa, archaea, lots of different things. We have mites living on us as well. And so I'm going to be talking about this other world that lives on us. So our body is their world. And so I think they're really cool, and we'll learn a lot about them. And they really influence our health and behavior. So pretty cool stuff here. All right, so when you think of the skin, because we're going to be focusing on the skin, so these microbes live all over you in different parts of your body. You've probably heard a lot about gut microbes, but I focus on skin microbes, and I'll tell you a little bit more as we go tonight. And so you can think of your skin, like I said, as your skin is your largest organ, and it has different environments on it. So just like our world has different environments for humans and other species, our body has different environments for these microbes. And mostly what I'm going to be talking about today are bacteria. And we know a little bit more about bacteria than we knew about some of the other organisms living on us. So like I said, I'm going to focus on bacteria, but you can ask me questions about any of the others. Most of my research focuses on the bacteria. And so we know that the bacteria live in different environments on your body. So as you can think of different environments in the world, there are some oily places where there's an oil slick maybe, or a nice tropical jungle, or a desert that has a really dry environment, right? So these different environments host different kinds of species on our planet. Just like if you have these same types of regions on your body, they host different microbes based on what lives there. Oh, and I'm advancing slides. How are we doing that? Okay. So you can think of your forehead has some oily parts on it, right? And so your armpit, which we're going to be focusing on, has a lot of moisture there. And then your forearm is really dry. And so each of these different environments hosts different organisms that live in these different places. And as I said, the bacteria we know a lot more about. And so if we look at bacteria, they actually live in different environments based on what the type of environment is like. But if you talk about fungus, what's really interesting is that they don't really live in the environments. They more are specific to the certain body region. So there's something a little bit different about bacteria and fungus at this level. And like I said, we're going to talk about the bacteria, but the fungus has probably a big role that we just don't know about yet. There's a lot more that we have to learn. And so these ladies maybe one day will be investigating some of that. All right. So I want to just sort of draw your attention to the fact that sort of hammer home that if you look at different parts of your body and you look to see which microbes live in these different parts of your body, you actually can sort of see a signature that if you look at which microbes live in a certain part of your body, that they all sort of cluster together. The similar kinds of microbes live in, say, your mouth, or live on your skin or your gut, okay? And so this picture is just showing you different swabs that have been done on different people in these different regions. And so the site or the environment on your body is actually clustering which microbes, which kinds of organisms live in those environments, okay? Okay, so why do I focus on armpits? How many of you guys have armpits? All right, how many of you want to be swabbed after the event tonight? Oh, wow. Only 1 or 2 takers. Wow. All right. Well, maybe by the end of this talk, you'll all be really enamored and you'll want to know more about this life. All right. So why armpits? Well, I started this museum about 4 years ago, and my background is actually as a comparative geneticist. So what I do is I study the DNA of humans and compare that to other primate species to really understand, well, how are human sequences different than other primates, and how might that influence our health and well-being? Okay? And so when I came here, we started thinking about, well, how can we engage the average museum visitor in some of our research? And while people like primates, right? You guys all are primates, so hopefully you like other primates. It's really hard to get scientists or citizens to actually be involved and collect data from primates, right? So how many of you guys would go to the zoo and spend 8 hours studying the chimpanzee behavior? Probably not too many of you, right? But if I now say, hey, would you come and could I swab your armpit for science? Now we actually get a lot of people who are dying to give me their armpit swabs. Okay? So we thought, well, hey, this is a really cool way to engage visitors. Why don't we look at the microbes that live on different people? And we can swab people for these different microbes. And we can look at humans and we can look at other primates. And if we think of these microbes, one thing I want you to know is your body odor is produced by the microbes that live on your skin. Okay? So you have all these microbes living on you. They feed off of the sweat and the oils that you're producing, and then their byproducts are their metabolic compounds that they're giving off, these metabolites that we call. Those metabolites are, one form of them can actually produce the body odor. And so it's not you guys that smell, it's your microbes that produce this body odor. And so when we think of primates, a lot of us probably think, well, they use scent to communicate. If any of you guys like lemurs, they're some of my favorite primates. They use scent to have stink fights, and the stinkiest lemur will win the stink fight. And so it's their microbes that are producing these odors, okay? So if we now think of humans, there have actually been some really cool studies where they have men wear T-shirts for, say, a week and get really smelly and play basketball for a week, okay? And then they give that T-shirt, the smelly T-shirt, to a woman. And they say, will you rate the odor of this T-shirt? Do you like it? Is it attractive to you? And it turns out that in a number of studies that have been done, if the woman likes the smell of the man's T-shirt, and you look at the immune system genes of the man and of the woman, it turns out that the woman who likes the man's T-shirt, they probably have very different immune system genes. So from an evolutionary perspective, this could be really valuable, because if you're going to choose a mate by scent, and probably a lot of you who choose a mate have actually had scent play a role in that, oh, this person smells really good, I'm attracted to them, right? So if you're choosing a mate by scent, and that actually tells you that you're going to choose a mate based on their scent, but then they're going to maybe have a very different immune system. If you have children, your children are probably going to be able to combat pathogens better, because you're going to have a more broad repertoire of, these immune system genes. So this, from an evolutionary perspective, is really important and interesting. And so having my primate connections, we thought, well, smelly armpits are the way to go, because everybody here has an armpit, right? I talk about smelly armpits, and people are like, ooh, oh, tell me more. That sounds intriguing. And so I can entice and engage people with some of that research, but then I can really tell you that this is really influential and important for your health and well-being. And we'll get into that somewhat later. But it's sort of a way to hook people, right? Hook people on the science and get them engaged. As you hear from all these ladies, they tell you, maybe one of the reasons that they're sitting up here is because I do armpit microbe research. They wanted to know more. And so that's a good way to hook people, but it has some really important evolutionary and health perspectives. And so I think that's really cool. So one of the first things we did was we swabbed people's armpits, and we just plate on a plate one of these Petri dishes. And if any of you want to see this, this is just a nice plastic Petri dish. And we put agar in here, which basically looks like Jell-O, sort of a Jell-O jiggler for the microbes. And it has food for the microbes. And so you put them on this plate, and you grow it up at body temperature, and different microbes will grow. And so you see a lot of different kinds of bacteria on here, sometimes some fungus. And so each person's plate sort of looks unique. So each of you are unique, but the microbes that live on you are also sort of a unique composition of which microbes live there, okay? So when we started in the lab, we thought, okay, well, we're starting here. Maybe we're going to start swabbing people's armpits, and they'll be really engaged and want to learn what lives on them. So what do you think happened when I swabbed my armpits? Do you think that I'm up here? Anybody think I have cool armpits up here? No? What do you think my plates look like, sir? Oh, you know the story. So we swabbed my armpits, we swabbed my belly button, and all the plates from me grew nothing in the lab. And so as a most of you guys were probably like, oh, that's awesome. I don't want microbes on me, right? They're icky and germy. But it turns out I was freaked out as a scientist, right? I should have microbes growing on me. They're supposed to be good for me. So where did they go? Well, it turns out I got this job, and I was talking to the public quite a lot. And I get a little nervous talking to the public. So I started using clinical strength antiperspirant. And it turns out that one of the reasons you use the antiperspirant is because you want to block your sweat glands so you don't produce a lot of sweat. And if you're blocking your sweat glands so you don't have sweat, there's no food for the microbes. So then the microbes don't grow on you, okay? And so I was using this clinical strength antiperspirant that was probably wiping out most of the life under my armpits. And as I took a shower, it probably trickled down and affected belly button microbes and maybe some other microbes, although we didn't swab other places, okay? So this kind of got us started thinking, well, what am I doing to my body if I'm wearing this antiperspirant that's really strong, right? So does this have any health consequence for me if I'm wiping out the life that could be really important for me? So this sort of started us on our first project, which was in the dead of summer here in North Carolina. So in the middle of August, we said, okay, well, who would volunteer to go without deodorant and antiperspirant for a week? How many of you would do that? A couple dedicated people. Okay, good. That's pretty good. Good, good. We actually have some dedicated volunteers in the audience here. Thank you very much. So we actually started this project to see, well, what happens if you ask somebody to stop wearing their normal product? What do they look like on that first day when they wear a product? And then what happens if you stop wearing product, okay? So what's going to happen to the life? How long is it going to take that life to grow back? So if I want to study the microbes, I have to have microbes there to study. And so people who wear deodorant and antiperspirant maybe aren't going to have any microbes in their armpits, so I don't have anything to study. So I'm going to need to figure out, if we do some of these projects, how many days am I going to have to ask people to stop wearing product before I can actually collect enough sample to study the microbes living in their armpits, okay? So that started our first project. And so what we did is we said, okay, come into the lab. On the first day, if you wear antiperspirant or deodorant, we're going to swab your armpits. We're going to plate them out on these plates and see what grows. And most people who wear product didn't have any growth on their plates. Then one day without product, you see here, left and right armpit, you're getting some more growth. So you're getting these little white colonies, which signify that bacteria are growing on this plate. And then after 2 days without wearing product, you get even more growth. And so it took at least 2 days for most people to get back significant growth. And then after 5 days, they had pretty much an even amount of growth as somebody who doesn't wear any product at all, okay? So one of the really interesting things about working with citizen scientists is I approach things as a scientist, right? So I develop a hypothesis from some of the data I see, and then I come up with this hypothesis, and then I want to test it. But we have a lot of people who come in and say, oh, you know what? I have this really interesting fact. My wife really likes the smell of my right armpit. You know, do you think my right armpit looks any different than my left? And it turns out, if we plate this person's microbes, that the right armpit has more microbes growing there. And it turns out that this right armpit is apparently smellier. So it sort of suggests that maybe there's actually a testable hypothesis here. And so I can actually get information from our citizen scientists, and that can direct some of my further research. And so that's what's really cool about having students here, because they come in and they're like, oh, Dr. Horvath, you know, did you think about this? Or, you know, I started using that. Maybe we could test this. And so we get a lot of interesting new ideas from all these people that we talk to, which is really fun and intriguing.
Julie, are you going to get a new paper out of my smelly right armpit?
That would be awesome, Brian. Would you contribute more sample to us?
Absolutely.
Awesome, awesome. See how dedicated everyone is here? We love this. All right. So what did we find when we did this study? So if you've seen any press, there's been some press about this publication recently. And what we found is that people who wear antiperspirant and deodorant actually have different microbes living on under their armpits than somebody who doesn't wear any product at all, okay? And all right, so we have these pie charts here, which are somewhat similar to the pie charts that you have. And what we do is now, instead of just culturing the microbes, we actually isolate all the DNA from all the microbes under your armpit. And then we sequence a piece of one gene, this 16S ribosomal gene, that's this barcode gene. So this gene is conserved among all bacteria and Archaea. So all bacteria and Archaea have this gene. And so we sequence this gene, and the sequence of the gene is specific to each individual microbe species. So we can look at the sequence of this gene, and then we can say, okay, let's look at public databases, and other researchers have actually determined which microbe species has which sequence of this gene. So we can look at our gene sequences that we find on people, and we can match it to the public databases and say, okay, now I know that this person has, most of the sequences we get back from this person match a Staphylococcus or match an Archaea bacteria, et cetera. And so we can sort of use our sequence now to really match and figure out which species live on different people. And so what you see from this graph, or these pie charts, is that if you wear antiperspirant or deodorant and you stop wearing that product, and 2 days later we sample you and look at the microbes that live there, most of what lives on your skin is a type of staph bacteria, okay? We only really determined it down to the family level classification, but it's some type of staph that lives on you predominantly. And you have a little bit of corynebacteria in green here. You have a little bit of Anaerococcus, but then you have a lot of gray and black stuff. That's just these other things that are really low abundance on you, but they're other kinds of microbes. Now, if you typically don't wear any product, you have the flip here. So in the small sample that we looked at, if you typically wear no product, most of what lives under your armpit is the corynebacteria. And these are bacteria that typically have a lot of body odor. They produce a lot of body odor. So it's interesting that people who don't wear product might have a lot of body odor, okay? And the other thing that was really interesting about this study is that if we look at the people who wear antiperspirant now, you see a lot more of these other microbes that live on them. And some of the people who wear antiperspirants contain aluminum salts. And some of these people have Archaea. Archaea are thought of as really extreme organisms. Some of them live in deep thermal vents, can really withstand high salt conditions, high temperatures, okay? So if you wear antiperspirant with aluminum-based salts, maybe you're selecting for Archaea bacteria that grow on your skin, these Archaea. And so what's really interesting is that maybe your daily habits now are really influencing the microbes that live on you. And just think, how could this be influencing my health and well-being, okay? So we might get in that at the end, or you guys can ask questions about it. All right. So I told you that I'm going to put this in the context of primates. So we looked at humans to see what grows on humans. So now we went and collaborated with the North Carolina Zoo, and we have some other collaborators in Puerto Rico at Duke and UPenn, and we got some of these samples from primates, non-human primates, and we looked to see, well, how do the microbes on non-human primates compare to humans, okay? And I'm just going to show you the plates here, because what you really see is that when you look at human plates, a lot of the microbes on them are just these little white dots, okay? And chimpanzees are our closest evolutionary relative. They have similar microbes in their armpits. Gorillas are our next closest relatives, again, somewhat similar. But if you look at baboons, which are old world monkeys, their microbes look somewhat different now. So we're starting to see some really interesting differences here. And what's cool is that we all have different glands under our armpits. And so these glands might be differentially feeding the microbes on us. And so maybe that's one of the reasons that we have different microbes on us than some of the other primates. But you can also imagine that our daily habits are probably very different than these primates, right? They don't take showers every day. They don't wear deodorant, at least most of them that I know don't. And so starting to think now about this context of, well, how different are humans than our closest relatives, and how might our daily habits be really affecting some of these microbes? All right. So one of the studies that we've done, so I told you that we did this study in the hot summer of North Carolina. And from that study, we learned that our daily habits, like our antiperspirant and deodorant use, really affect the microbes that live on us. So I wanted to really explore how the microbes might be correlated with body odor. Okay, so I wanted to go back and do the t-shirt study. And so this past summer, with a lot of help from Sarah Council and a lot of other people in the lab, we went and we studied, what happens if you take people and you ask them to stop wearing deodorant and antiperspirant and to use fragrance-free products and then wear a t-shirt for a couple days at night to bed. And then after 3 days of wearing this t-shirt, would you bring that t-shirt back in? And can we have a smell off in the museum where we trade t-shirts and we let other people rate the smell of your t-shirt? And this actually came about, I don't know how many people know this, maybe a couple, because when I was in graduate school, there was one particular gentleman who smelled very, very good. Everyone thought he smelled really good. And we used to think it was, the whatever products he used. But I kind of wonder now if it was the microbes that lived on him. So what I was hoping from this experiment was that we would do this study and there would be one t-shirt that everybody thought smelled really delicious. And then we could look at that t-shirt, figure out which microbes live there, maybe figure out which metabolites are given off by those microbes. Maybe we could market that as some new fragrance, right? So that would be really, my husband thought this was the best idea ever too. Unfortunately, we did not find that, and I can talk a little bit about our t-shirt results. But this is sort of setting us up to think about, well, okay, can you guys rate t-shirts? Okay, so how many of you think that if you smelled your t-shirt, that you would be able to tell which t-shirt was yours among 20 other t-shirts? You think you could find your own t-shirt by smell? What do you guys think you could do? Could you do it? Wow, only a couple of you. Really interesting. I thought everybody would be able to know their own smell. So let's see. So we did this large citizen science project again, and we, collected data from, actually this was about 25 people. And so we collected microbes from people. We collected, we got them to wear t-shirts so that they would get this nice fragrance, their own body odor on the t-shirt. And then we also collected metabolites. So these are these small molecules that are given off by potentially us and the microbes. So we can collect some of these molecules and we can collaborate with wonderful people like Susan Sumner at RTI and try and figure out now which microbes do we have living on us and which metabolites might we also find on our skin, and then how can we correlate these? Can we figure out which microbes are maybe producing certain metabolites, okay? And so we had this speed smelling event to do this wonderful t-shirt test. And it was really awesome in the museum. We had a lot of good participants. And so we had people, like you might recognize this woman, smelling one of these t-shirts. And it turned out that people really didn't mind smelling the t-shirts. They didn't smell as strong as we thought they would. And so we had a lot of fun with this event. It was a quick little event, and people rated the t-shirts for us. And so what I thought was really interesting, we had 23 participants who wore t-shirts for our study. 20 people smelled those t-shirts. And then 14 people were both participants in both parts, gave us a t-shirt after wearing it for 3 days, and then also smelled the t-shirts. But only one person was able to identify her own t-shirt, which I thought was really crazy. And that person must have a really good nose because she also picked out somebody else's t-shirt and said, I think this is so-and-so's t-shirt. So she is an awesome smeller. And so we have some really great people who work here and work with us. So thank you very much for that. And so I did say there were 4 t-shirts who smelled good by, a lot of people rated them as smelling good. But like I said, I was looking for this one t-shirt that was like our gangbuster. This is the best smelling t-shirt ever. We didn't quite find that. We found a couple that smelled good. We're still in the process of analyzing a lot of the data here, but what you have on your tables are actually people's samples from this project, okay? So I said on day 1, these people came in and they didn't do anything different. So what do their microbes look like on day 1 when they go about their normal habits? And then after 3 days of using fragrance-free products and not using any antiperspirant or deodorant, now if we look at their microbes, how different are their microbes, okay? And so depending on what category your person was in, so some people wore antiperspirants typically, some people wore deodorants, some people wore no products at all. So everybody on their table probably has a slightly different pie chart. So you can ask me questions about that at the end. But it's kind of interesting to see who changed and who didn't change so much. Okay, we can talk about some of that. But one of the current projects that we're doing and why these lovely ladies are sitting up here with me is because they're starting to analyze data from one of our newer projects. Okay. And so how many of you guys know what kind of earwax you have? If you have wet earwax or dry earwax. Do you know if you have wet or dry earwax? How many of you knew there were different kinds of earwax at all? All right. So if you have dry earwax, it's probably flaky and whiter. If you have wet earwax, it's more likely yellowish, brown, and sort of sticky and gooey. Okay? So earwax is produced by this apical gland secretion. And there is one gene variant that actually controls whether you have wet or dry earwax. So there's one single nucleotide polymorphism, one single base pair in your genome that controls whether you have wet or dry earwax. And it turns out that a lot of Caucasian and African American people have the wet variant. There are some that have the dry, but very low levels. A lot of Asian population has this wet or the dry variant. Sorry. So in Japan, this was actually studied for quite a long time. There have been some papers looking at this proportion. And it turns out that people sort of thought it was silly. Well, who cares if I have dry or wet earwax? Why are you putting all this money into it? Turns out this gene is an efflux pump. So it can move things in and out of cells. And it turns out that whether you have the dry versus wet variant determines how you metabolize certain drugs. So now they're starting to find that whether you have the dry or wet variant determines how you metabolize drugs if you get chemotherapy after breast cancer, or how you metabolize some other drugs in your life. And so this could be sort of getting on the personal medicine realm, where what your variant is in this gene could actually be dictating how you respond to drugs and how your health goes, okay? So what's really interesting is now you can think about this. So we talked about these different environments on your body, right? So if you have dry earwax versus wet earwax, just imagine that that's probably a different food source for different microbes. So maybe whether you have wet or dry earwax determines which microbes live on you. Okay? It's not too outlandish. Your daily habits influence which microbes live on you. Maybe your gene variants also at some level influence which microbes live on you. So what we're doing is looking at collecting samples from people with dry or wet earwax, and actually we're looking for people with dry earwax. So if you have dry earwax, I would love to sample you. Please come up and talk to me. We're looking for a few more wonderful volunteers for this project. You know, these lovely ladies are going to start analyzing some of the data, so you can talk to them about what some of the things that they're finding. And so we're looking to correlate the microbes that live on your skin and your ears and your armpits and see how that might change whether you have dry or wet earwax. Okay? So before I get too much farther, so I'm going to sort of wrap up my part of the talk right now, and I'm going to hand it over to these ladies so they can tell you a little bit about what they've learned. But I wanted to say thank you to all of you and all of our wonderful volunteers, and our non-human primates, of course. All right. And I've got to give a good shout out to Bobby Reagan, my NC Central grad student, and Sarah Council as well. Gets them set up. All right, so I'm going to let Ms. Tamara Allen come and talk first, and she can talk about some of these microbes that you see on this plate.
Hello, my name is Tamara Allen, and I attend North Carolina Central. Last semester, I had the opportunity to take an evolutionary medicine course with Dr. Horvath. Overall, that class really made me look at how we get sick in a different perspective. And I had the opportunity to also volunteer in the citizen science project as well, where we swabbed my ears and armpits. And on the screen, that is my left ear and right ear. I had to follow a protocol where I could not wear deodorant for 2 days, and I couldn't shower before class, and I couldn't clean my ears for a couple of days. So this is what grew on my ears. The next one is my armpits. The right armpit seems to have a little more growth than the left. I don't know why, but that's pretty interesting. So from seeing these plates, I really wanted to learn more about what was on the plates. So since I've been working in the lab, I realized it's not that easy to classify what is on the plate. And I've really been learning a lot. I learned how to take pictures of plates, and I learned how to make the LB plates that we use to grow the bacteria. And I really found that it was interesting that corny bacteria is high when staphylococcus is low. And I found that interesting because I know that everyone has staph on their skin, so I just generally thought that it would be high for everyone. And I thought cornea bacteria would be high for people who have more odors, but that didn't seem to be the case. So I'm really having a good time learning and actually trying to put these things together and realize that our everyday habits do actually have something to do with what grows on our body and could essentially be something that contributes to our health. So it's really what I'm learning, and it's really interesting, and I hope that everyone else finds this pretty interesting and will help us to help volunteer and, you know, give us some samples so that we could further, you know, investigate this thing, because it might be something that's really big. So thank you.
Thank you.
Hello, my name is Nina Pettis, and I'm also a student at North Carolina Central. I decided to join Dr. Horvath's lab because she looked at the microorganisms under your armpits. And I found it interesting because I get a chance to use my body in science. So you don't get to do that every day at school. So you're always following protocol because of class, and 50 other students ahead of you have done that already. So I found that pretty interesting. And right there is my neck. So it's pretty gross. There's a lot of yellow stuff up there. So I'm not sure what that is yet. I haven't got a chance to analyze it. But I got to swab my neck, my arm, my belly button, and my leg. And I found it interesting that my belly button didn't have that much growth on it, and I thought it would because, you know, your belly button is kind of gross, and I have an innie, and you would think you had to get deep in there and find some good stuff. And also found interesting that the type of deodorant I used to use Dove Solid, and it only stopped odor, but I continued to sweat. And now I use Mitchum for women, and it stops both odor and sweat. So I haven't got a chance to swab my armpits yet. So when I do, so maybe I can find out what organisms grow under there, and maybe I can find the best type of deodorant for me.
Hi, everybody. My name is Dawn Stansel. I'm a senior at North Carolina Central University. I am very excited to be here. I was looking for a research opportunity outside of the school, and I heard someone was swabbing armpits. And so I thought, okay, armpits. Then I heard about earwax, and I said, okay, why? And then I thought, this is intriguing and quite eccentric, so naturally I'm in. My level of curiosity has always been extremely high since I was very small. My mom, when I decided to pursue this career, said, I should have known it. You've been asking why since you were 3 years old. So as time has gone on, I've worked in the lab over the summer, or over last semester as well as this semester, and I enrolled in the research course because I wanted to learn more. I've had an opportunity to swab some of these lovely microbes that you all have so graciously given us, and I've also had the opportunity to see what grows on them and take a few pictures. But what we're looking at here is actually a sample of the back from the middle, and you can see there's probably 9 or 10 different dots, if you will, that look completely different. And from what we're finding out, there's thousands, hundreds of things that are living on us, and we're trying to figure out how they grow, what they're doing there, and if they're beneficial to us or not. But there's some interesting things about what's on that plate. If you guys can see to the upper left-hand side, there are circles, but there's also a little line between that circle. So that means to tell me that for some reason, whatever's on the top doesn't really like what's right next to it, and so they've kind of created a barrier. And that, I think, is going to further our research in trying to figure out what these microbes do, what their purposes are, and/or health benefits. So I just wanted to tell you guys that I enjoy working in the research lab, and I'm here sometimes for 5 hours, and it truly feels like 5 minutes. So that's how I know I'm exactly where I need to be. Thank you.
All right. Thank you very much for Julie Horvath and all her students. And were you about to say something? No, you're done there. Excellent. So yeah, that's very cool. Not only are all these things living on us, but they're fighting, and we don't even know what they're doing, right? So now the rest of our program, as most of you know, is Q&A. So we're going to open the floor to questions. And you can ask Julie anything, of course, but feel free to ask her students. And by the way, these are undergrad students. They're not grad students. They're not already scientists. But these are undergrads that took a class with Julie, right? Am I right?
You are right.
So, yes.
Microphone, dear.
Yeah, we're gonna. So all of our programs are streamed live on the internet, and so I will be bringing a microphone around to you and asking you to speak into it.
Okay. Well, deodorants and antiperspirants have been around for decades and decades. So, have you looked into the historical records of the peer-reviewed literature, what did all of these consumer health companies do that enabled them to put a deodorant or an antiperspirant product on the market? And were any of the things that you're looking at, were any of those things part of their considerations?
So that's a good question. We haven't, been deep into that literature, but a lot of these products are meant to stop the growth of microbes, and so that's what they do. And sometimes they might have negative health effects, but you need long-term studies for a lot of those, and sometimes we don't always have the information for that. So I know there have been links suggested to certain problems, certain health concerns, and none of those have been verified that I'm aware of. And so I think, these products are still out there, and I want you guys to think about what you're putting on your bodies every day, because doing this study really made me think about that. And so it's a good question, and I think it's something that we just don't know yet. A lot of the studies that were done even 10 or more years ago were done with culture-based systems. So we can grow the microbes that live on you, we can grow those in the lab, but we're missing a lot of what lives on you if we try to grow it in a lab. And so I think now we're starting to learn a lot more now that we can actually sequence these microbes, because now we can look at pretty much everything that lives on you. And so I think we're going to start learning a lot more about some of the health benefits as we do some of the larger studies.
Julie, have you actually changed anything because of this? You said that it's made you think a lot about it. Have you changed anything?
I have. I've certainly experimented with a lot of different products, underarm products. And, deodorants usually contain products that kill microbes. And so those don't work as well for me. You still can sweat. And because you can still sweat, you can still have microbes growing there. And so I've tried deodorants. I've tried natural products. I don't use the clinical strength anymore, but I do still sometimes use the regular antiperspirants. So, yes, I have changed.
Do you all study differences not only between male and female, but old and young?
So those are really good questions. So males and females have, there are significant differences between males and females. So the underarm product seems to be a really big factor in what grows on you, but male and female potentially overshadows that. And age definitely also makes a difference. Not so much maybe on the skin, but for gut, it's certainly your age can change. But, I've heard the old people smell bad rendition is actually somewhat true if you look at the metabolites and the smelly compounds given off, that older people do actually produce some byproducts that have a different odor than people who are younger. So people over 40 maybe actually do smell differently than people younger than 40. So there are some age differences there, likely contributed a lot by the microbes. And so that's one of the things we could look at with more data.
Hi.
Hello.
I'm a microbiologist by training, so I don't want to share my life with you, but I am going to ask about this rationale. I don't like antiperspirants. I don't like antimicrobials. So I use isopropyl alcohol. Since corynebacteria, I think, create fatty acids that sort of keep other microorganisms down. If on a daily basis you knock down the population that's all on the surface, the natural organisms are still in your pores. Even when it's summer and you get a little gamey, if you do it on a daily basis, it seems like it works fine. What's the benefit of doing that and preserving your natural flora? The alcohol, I assume, lets the natural flora come up with the antimicrobials, changes the mix. Mm-hmm. What's the advantage of doing that?
So that's a good question. I think that's one of the things we're still trying to learn. And so I'd love to talk to you more, but I think we still have a lot to learn in this realm, and I don't know the answer to that.
It's a good question then.
Yeah. I was wondering if you were able to correlate different microbes with discernible different fragrances, shall we say, and if those fragrances, odors can be different on different people.
So good question. So one of the things we're doing now is analyzing some of the data, looking at which microbes live on you and then which metabolites are found on people. And so we haven't done that correlation yet. That's something we're actually meeting with our collaborators next week, in fact. And so that's something that I would like to do. There are certain people who have, in the lab, taken certain microbes, grown them on plates to try and figure out which compounds they give off. There's a really interesting piece with mosquitoes, because mosquitoes actually cue into the volatile chemicals, your smells, and so potentially there are some microbes that give off different smells than others. So some types of staph have actually been linked to being more attractive to mosquitoes, and so I think there is something with that, but with our data, we don't have that information yet. We're still analyzing it.
Yeah, I noticed that you said when you use antiperspirants, there's a higher percentage of staphylococcus, and that's when I associate with skin infections with staphylococcus. So do people that use that type of deodorant have an increased number of infections or severity of infections?
So that's a good question. So the way that we did that study, we had people where, the people who normally wore antiperspirant, we asked them to stop wearing. And so it turns out if you stop wearing product, certain microbes grow faster than others, and the staph grow faster than some of the Corynebacteria and some of the other microbes. And so potentially we were seeing that effect was those are the microbes that grow back faster when you stop wearing product, right? So if you can think of, you have this beautiful forest and a fire comes through and wipes out all life there, that's kind of equivalent to using an antiperspirant on your skin. And then after the fire leaves, now what grows back the fastest? Well, some of the weeds are going to grow back fastest. And so what grows back on your skin after you stop wearing product, after you've wiped out life? Well, the staph are the kinds of bacteria that grow up the fastest. And so that might be a byproduct of some of what we looked at. But there certainly could be health implications down the road, because some staph, we know that staph grows all over your body, and so some of it is presumably good for you at some level. If your microbes get out of balance, so like I said, you have bacteria on your skin, you have viruses and fungus on your skin, and sometimes these microbes are actually in check with each other, and so they're when they're at a healthy level, they're good for you at that level. But if one of them gets out of whack, then that can cause another one to cause a problem. And so that is something that we're considering looking at down the road. One of the studies that we did, we were looking into the literature, and it turns out that a lot of athletes get staph infections in their armpits. And maybe that's because they wear product and maybe with all the sweat, it sort of causes their staph to grow back faster. And so maybe that's one of the reasons they could get some of the staph infections. But some of the staph are good for you. Certainly some are pathogenic and cause disease. But some of the microbes that live on your skin are probably pathogens, and they're not doing anything to you right now. But if you perturb the balance, then that can cause a problem. So those are good health questions, but there's just a lot we don't know yet.
Sounds like it's very complicated.
Yes.
So we may all be talking about the same thing, but I work in healthcare on the communication side, and one word I hear a lot of times is superbugs. We're hearing it a lot on the antibiotic side. Is kind of what you're saying of the clinical strength things, or even what you're just talking about now with athletes and staph infections. Is that kind of on the similar wavelength of we're killing too much in our armpits and maybe not what's supposed to be there isn't there anymore, like the superbugs when it comes to antibiotic overuse?
Yeah, potentially. I mean, our daily habits are affecting the microbes that live on us. We know that there's something called a hygiene hypothesis, which actually posits that not so much hygiene's how often you wash your hands, but hygiene's in the sense of sanitation. So we have, flush toilets and a lot of things that are more clean as we think of them, but our bodies we co-evolved with some of these microbes to live on us, and some of them actually do things that are good for us. So we're actually losing some of these microbes that could potentially be good for us, and so all of these daily habits are maybe playing a role in that. And so we might get asthma and immune infections more often because the microbes on our skin really train our immune system, and so if you're perturbing that balance, you could be changing things that potentially were good, and now you've done something to that mix. But again, there's so much that we don't know about what's healthy, and I think that's where the primate research comes into play, because we look at all these different people. We could look at 10,000 different people and say this is what a healthy skin microbiome looks like, but until we put it in the context of, well, what do our closest evolutionary relatives look like? What does their skin look like? How much staph is on that organism? And so I think that's starting to put us into context with some of this work. But you're right, we could be selecting for some of these things with all of our daily products, certainly.
Hi, I just wanted to ask, have you done much research on different nationalities of people eating different diets? Like, for instance, people that would eat more like curry and things like that in their diet, or, Asian people that eat different types of foods? like, and how that would affect their microbiomes, and of course, it would affect the smell, I would think, of how they, what I would eat versus what they would eat.
Yeah, so that's a really good question. Right now, we don't have a large enough data set to look at that. We haven't specifically focused on different ethnicities. There are other studies looking at the oral microbiome and the gut microbiome, suggesting that your ethnic background could influence which microbes live on you. So if you look at a particular, if you look at Caucasians versus Asians versus African Americans, you can actually see signatures based on what your ethnic background is. So at some level, that's probably getting back to our host genotype, right? What genes we have and the variants of our genes in our body are potentially influencing which microbes live on us. So even though my research hasn't touched on that, a lot of other research has, and we are seeing some differences. So there are a lot of things that play roles there, and your diet could be one of those differences. It could be based on, your cultural basis, or maybe just the kinds of foods you eat. So we do ask a lot of our citizen scientists questions. Now we're actually up to asking people 60 different questions, and some of those are diet-related questions, and we don't have enough data at this time to answer that. But I think the diet probably does influence it at some level. But there are a lot of things that influence your microbiome, and so each one is a little piece of the pie.
So 2 quick questions. The first question is, for ear infections that kids get all the time, does it matter if you are dry wax or wet wax? I'm just curious, is one more susceptible to ear infections than the other? That's the first question. And then the second question is for one of the students. So I think one of the students had more on their plates under their arms. They had more bacteria under their right arm versus their left arm. I'm just curious if that student is right-handed versus left-handed, and could that have made a difference? Because your dominant hand, you may do more on that side, which may cause more sweat. And just curious.
You went first? Go ahead.
So, can you go back to it so I can see what yours was? Okay. So, I am right-handed, and the right armpit does have more, but also, so I shave my armpits, but one seems to have like more growth than the other one. So I don't know if that has something to do with it, but I think that it could have something to do with it because they look similar, but one does have more, so the right arm does grow out more hair, so maybe that could have something to do with it.
And I'll add, Julie had earlier in her presentation, she showed a couple slides that were cultures from my armpits. And going into it, I had told her that I knew from my wife, I'm right-handed, Antonio. And my, as often is the case with mates, my wife likes my smell, even if I'm kind of, if I've sweated. And so, but it was always the right armpit, always was more pungent than the left. Just like if she'd check it out, she'd go, yep, that's the smell. And over here, not so much. And then it turned out when we cultured them, the left, the culture didn't have that much on it, and the right had a lot more. So we knew I am right-handed for what that's worth. And that's the one that always tends to smell the most, and it had the most bacteria in the culture.
All right.
And then there was a second
I was going to answer his second question. So you're asking about ear infections, and I don't know the answer to that yet. We're still compiling a lot of data from our citizen science project, but one of the questions, or some of the questions we asked people were, as a child, did you get more ear infections than the average person? Do you get more ear infections now? And so we're starting to look at which microbes live in the ears of some of those people to see, do they have a different composition of, which microbes live in their ears, and can we actually maybe correlate that? I don't know our number will be big enough, our sample size will be big enough, but those are really good questions to see, are you predisposing yourself to some issues, and is there anything you could actually do? So anecdotally, I do know someone who said he got a lot of ear infections in one of his ears, and the other ear was fine. So he took earwax out of the good ear and transplanted it in the bad ear, and has cured his ear infections.
Really?
So, thinking about things like fecal transplants and some of these other things that people do, that's totally anecdotal. So I'm not advocating that in any way, but just suggesting that, there are some things that people are maybe starting to try and learn about that maybe work for some people. I don't know if that would work for everyone, but I'll get back to you on.
Could you elaborate on that example of what do you mean about fecal transplants?
Sure, Ryan. Thank you for that.
It is one of the most fascinating things, and
it's really awesome.
Yeah.
So some people can have really problems with diarrhea. If you go into the hospital and get put on a lot of antibiotics, that perturbs your overall flora. And once that imbalance happens, then certain microbes can actually take over because let's imagine that you take antibiotics and you wipe out most of the microbes in you. Then some of the maybe pathogenic microbes can take over. Some of those, one is called C. diff, and that can take over in your intestines, your gut, and that can cause really horrible diarrhea. I mean, for months and months, people can have really terrible diarrhea. And one of the main things that they used to do was use stronger and stronger antibiotics to treat then the C. diff infection. It turns out that C. diff is one of these things that we could call a superbug where it can be resistant to a lot of different kinds of antibiotics. So now you're trying to wipe out even more life because you're trying to wipe out this C. diff. Well, what they started doing, they started a trial to actually take microbes from the feces of healthy volunteers and transplant that into people who had these C. diff infections to see could now good microbes actually take over and cure this C. diff infection, because could the good microbes now potentially, or beneficial microbes now overpopulate and take over, and C. diff would now lessen. And it turns out when they were doing this study, they had 3 different groups. They had people who got the fecal transplants, people who got stronger antibiotics, and I think there was a 3rd group that had some variation of that. And after doing this project for just a very short time, it turned out that the people who got this fecal transplant from a healthy volunteer recovered from their C. diff infection so much better than people who were on the stronger antibiotics or this other treatment, that they stopped the study and they said, we're going to start doing these fecal transplants. And now there are actually a lot of people who are marketing this as a new company. So there are companies starting out now to make synthetic forms of this concoction. And what's really interesting is we don't have long-term data on this, right? So this has just been happening over the past couple of years. And so I think we've been focusing a lot on the bacteria, like I said, but there's a lot we don't know about the fungus that lives in us. And so potentially we maybe need to add some fungal components to this and really learn a lot more about what we're doing with these transplants. And so it's really interesting to think about, but again, there's a lot we don't know about some of these other forms. And so it might be curing some people, but maybe there are some longer term consequences we aren't thinking about. I don't know the formal where it is right now.
I have 2 questions.
Okay.
I'm not a scientist, so all the bacteria that you are swabbing is external. It grows on the outside of your body. This is not excreted or transferred from inside pores or mouth or coming out of organs. It's all external bacteria?
So we are swabbing only the skin, correct. And so, like the skin on the outer side of your ear or the skin under your armpits. I mean, other people collect samples from many, many other places, but we are only collecting from the skin.
But it's not coming from the inside to the outside.
I mean, it could be coming from inside some glands in your skin, your pores, et cetera. But, most of what we're collecting are probably just on the outer surface. And so one of the things you have to consider is that, maybe I just brushed up against this table and then I touched my armpit. And so maybe I transferred some microbes from this table to my armpit. And so maybe I'm getting some of those as well. But presumably I'm just getting things on the outer surface of the skin. So yes, they are not coming from elsewhere. They're coming from the outer surface of what's on your skin right now.
Okay, the other question is, I don't use antiperspirant or deodorant. I use talcum powder. Can I assume that's just a drying agent and that's why it seems to work? Or is there something in talc that also...
presumably...
do they use talc in deodorant and antiperspirants, or is it just by itself an agent that would kill bacteria?
So I don't know if it kills. I know certainly the drying potential. So if you're eliminating the sweat, then there isn't the food source for the microbes. But I don't know enough about what's in talc to know, could it be antimicrobial at some level? I don't know if you know, sir. I don't know the components of talc, no. But, I have talked to a lot of people now who use very different things under their armpits. Many people have emailed me after we published some of these papers saying, oh, I use this product or I create this, et cetera. And so it's really interesting to think about all these different things that are more natural and what they do, and I just don't know because we haven't done those studies yet.
Excellent. This has been great, Julie. We're going to take 2 more questions.
Thank you. It's not really a question because my questions got answered, but I think we're just in the very beginning of understanding some of this, and the preliminary research showing how our gut flora is so important and it helps our immune system and what you're finding out. So I guess I just want to encourage the students up there to really keep pursuing this, because I think this is going to lead into really important science in terms of health and immune systems, and instead of using antibiotics and all these other things. So I just want to encourage you to keep going with what you're doing, because you could end up being leaders in this field, because this is still very new, and it's going to be, I think it's going to be really significant. So I just want to give that encouragement.
Yeah, thank you. Anything to add to that, Julie? Or?
No, I totally agree. I mean, I've really, actually, teaching this course, this evolutionary medicine course, you know, it got the students to think in an evolutionary perspective about some of these diseases. But the thing that I really enjoyed about the course most was having this citizen science component, where I got some of my students to participate in this project and really see science in a different light. Because now I wasn't just telling them things, but they were participating and learning about the microbes that live on them. And I actually saw a spark in some of the students who didn't necessarily like reading the papers and talking about what they learned in a paper so much. But then when I started talking about the microbes, and they could look at this, you know, huge Excel sheet of all these different microbes and how much was on each person, like that sort of sparked this new idea in a lot of these students. And so I think that was what was really intriguing to me, and that's why I have them up here, because they've all been really awesome to work with, and it's really been rewarding for me to just see, oh, I can like expose them to new things that gets them really excited about science.
Excellent.
Yeah, sorry, probably going backwards here, because that was a good finisher over there. A quick question. We always hear that the microbes last about 7 days on surfaces, but how long do they last on human body?
That's a great question. I don't know. I know in the sense, if you look at your skin, there can be a turnover of your skin cells in about a month, about 30 days. But I don't know how long they live on human skin. You know, different microbes, I'm sure, live different amounts of time. If you grow them in a lab setting in a dish, you know, you can grow them and have them on a plate over the course of a week. Certain ones will grow up. Some will grow up faster than others. Then you can put those in a refrigerator, and, you know, they'll go dormant for a while. So some presumably could be on you, and I just don't know how long, you know. And one of the things I was mentioning, when we swab on your skin, we're swabbing whatever is there. And so some of it could be alive, some of it could be dead. Probably most of it is living at the time, but some of it could have died, and we just are swabbing it now because we're sampling it. So it's a good question. I don't know that we really know how long they all live on the skin.
Interesting. Well, Julie, this has been fantastic. Well, maybe we could take one more question, and that's it, though. We're gonna have to wrap it up. But let me come on over here and. Sorry?
Loud enough.
Not loud enough to be heard on the internet, I don't think.
The staff.
Yes.
You haven't said what types. Have you actually learned any particular types of staff that you have found on off of anywhere on the skin? And in particular, have you seen any of the staff that can cause sepsis? Are serious infections that can be life-threatening just in your swabbing?
So great question. Dawn, do you want to start with an answer, and then I'll pick up?
We've actually found very large amounts of Staphylococcus aureus, and that's one of the main main species. But there are about 41 to 43 different species of staph. And so we're continuously finding out what we have. And it's kind of hard to get it down to the species level because they are similar in their
the gene
genomic.
Yeah. So actually, so one of the studies that we did, we were looking at this small piece of one gene, and with that we can say that it's a type of staph, but we don't know exactly what the type is. And with some of our other studies, we've been able to say, well, let's get down to the genus level. It's a Staphylococcus, but then I don't know the species level right now. And so with Staph aureus, even though if you can get down to the species level, there are different strains, and some of those are the MRSAs you've heard of, the methicillin resistant, and some of them are not. And so we certainly won't ever get to that level with our research, and that would be another specific assay that we would probably have to do. So we could maybe get down to the species level, but it's unlikely with the research that we're doing right now. We're more at the higher levels with some of this. It's a really good question. And you know, when we grow things on plates, you can sort of look at what the colony looks like and determine maybe what it is based on what it looks like.
Well, Julie, thank you very much. This has been great. We've covered a lot of interesting questions and topics. What do you want us to leave here knowing? Maybe, I don't know if there's anything that we didn't touch on, but, you know, like, why should we care about this? What do you want us to leave here knowing?
Good question. So I want you to leave here knowing that your body is a wonderland of awesome things living on you, and your daily habits influence the microbes living on you, but a lot of these are really good for you. So don't think of them as pathogenic or negative. Most of them are doing really good things for you. And so just think about your daily habits, how you might be affecting these good travelers on your body. And so, you know, that's one of my big take-home points that I think that's really interesting. I didn't think about that before. I really thought of them as this bad thing on me. It's a germ, but it turns out that they're mostly doing really good things for you. So you should protect your healthy biome. And I'd really like to thank all my students up here. They've been awesome up here today, wonderful to work with, and I hope you come visit us at NC Central.
So thank you, Julie, and your students. As I said, next week is Science Trivia Night once again, and this building's open till 9, so you don't have to leave immediately. But thank you very much, and we'll see you next time.
Awesome.
Good night.
If anybody has dry earwax, please come talk to me. If you want to contribute.
Excellent.